So hello everyone. So we are here with Dr. Nikki Fried, and I reached out to her because of the Midnight Protocol, which is very interesting, and yeah, this is just going to be a conversation about that protocol and to see whether um, we could apply that as well for our situation here in the Philippines. So, Dr. Fried, welcome to this this Q&A.
Thanks. Yeah, thank you for having me. I just saw online that you had put you have a crowdfunding initiative to try to get money for trying to get more accessibility to COVID genomics, and that's something I'm really interested in personally. And I just wanted to also highlight, I saw that you're using the ARTIC method potentially to do that, which is, I think, great. But I also just wanted to point out too, especially for labs that might be new to nanopore sequencing, using the rapid method, the rapid kit, might be a little bit easier than the ligation kit, which is what the ARTIC method uses. So that's the method that we had developed, and it's now on the Nanopore site. And so I thought I just wanted to highlight that to you, that might be of interest. As something that might be easy to adapt. And I have a couple of slides just to explain the difference between the two, for people who might not know what the difference is. And I'm happy to talk about that if you want, or just answer any questions, or just keep it free flowing, I guess, however you
Cool.
want.
Actually, personally, that's my question, because there's like different levels of technical depth here. I'm the I'm the most shallow in terms of technical depth. So, but my main concern is like, what is like if I'm going to explain this to like a local government, it's like it's okay. This is going to be the protocol that we'll be using. So, how would we explain that? Like, what's the main difference? So maybe this is a good time to look at the
Yeah.
slides.
Yeah, can I share my screen real fast? Yeah, let me do that. And then I can just take you through just real simple, the difference between the two. And it's a real quick, I just updated some slides for this. So let me just go back. I think you are all fairly familiar with what nanopore sequencing is, so I don't really have to talk too much about that. Obviously, the nice thing about it is it's portable and it does these long reads. For COVID, the long read aspect is not that important, but the main salient feature, I think, for COVID genomics is that it's portable. It's basically, there's no overhead cost, so you don't need a big, NovaSeq or MySeq or something like that. So it's fairly inexpensive in terms of the fact that you just pay for consumables. So that's why I had sort of reached out, is because now Nanopore just fairly recently made the rapid kit. They expanded the number of barcodes you can use. So previously, about a year ago, when Olin Silander and I developed this method with some other people, we were using the rapid kit, which only had 12 barcodes. So the difference now, why this is becoming a little bit more important or interesting, is that just in March, the Nanopore group expanded this to 96 barcodes. So now this means you can put 96 samples on one flow cell. So that brings the costs down considerably and makes it easier to use for lots of samples. So I just wanted to highlight, it's on their site now as, it has the fastest turnaround time, the least hands-on time, it's the lowest cost per sample, and it's good for people who want to incorporate automation. So that's what I'll be talking about today, is this sort of what we call the Midnight protocol. Oops. Yeah, so I think also most people know, so the ARTIC method, when people talk about that, that is what Josh Quick developed early on in the pandemic. So I think that the first genome was released on the 12th of January, and by the 23rd of January, Josh had released this really amazing, I mean, by no means do I want to say that it doesn't work. It works great. It's probably the most utilized method for sequencing the SARS-CoV-2 genome is the ARTIC method. And what we did was we just did some slight tweaks to this method to make it a little bit faster and simpler to use. So he should take all the credit for this. So what their method does, what the ARTIC method is it takes the genome of SARS-CoV-2, it's 30,000 nucleotides long, and you do a RT step, a reverse transcription, so you change the RNA to cDNA, and then there's a multiplex PCR. So I think also most of people here probably know this, but just to make sure we're all on the same page. So basically, then you put about 96, 98 primers in 2 tubes and amplify the genome in little 400 base pair pieces. And you do this in 2 tubes. So that's what I've sort of represented here, is just 2 different tubes with basically the whole genome covered in 400 base pair pieces. And the thing about the kit that they use to get the DNA ready for sequencing is they use a ligation-based kit. This is called the LSK 109 kit, and that takes about, this is from the Nanopore website, so it takes, on their site, it says about 135 minutes. It does a end repair step, which takes, it needs enzymes, usually purchased from NEB, so it needs some external enzymes like this FFPE repair, and then also a ligase to get these adapters onto the DNA and get it ready, prepared for sequencing. Okay, so that takes a bit of time, a bit of external reagents, and if you haven't done it before, it can be, it's a few more steps than what we have in our method. Okay, so then our method, so then the results here, so they had 3 different versions. Currently, I think they're just about to release version 4 of the primer set. But what you can sort of see and what people have shown is that there's not, again, I don't want to be critical at all, but there's not, because there are so many amplicons, they don't always perfectly amplify the genome in the same way or sequence in the same way. So it's not a real even level of coverage. So sometimes to get some, especially around the spike gene, which is of interest, a lot of times you can have these dips in coverage, which means you have to sequence more and more to get the whole genome at the right level of coverage. Okay, so that's an issue, that is sort of an issue with these primers, the ARTIC method. Okay, so we made 2 tweaks, I would say, to the ARTIC method, which is to use longer amplicons. So it's 1,200 base pair pieces instead of 400 base pair pieces. And the nice thing about this, at least in our method, we get a bit more even coverage of the genome. So the DNA or the RNA is effectively amplified more evenly. And because you have fewer primers to do that, it's a third of the primers, that means that as the virus is evolving, and mutations sort of start to appear and fix, with fewer primers, then you have fewer mismatches with the primers to the genome. So potentially using fewer primers is a little bit better as the virus evolves, so that you don't have as many dropouts. Okay, and the second little tweak we did was use what's called the rapid kit from Oxford Nanopore. It's the RBK004, or now they've just released RBK1110, which has, it's a transposase-based kit, and that makes it really simpler and faster and easier to use. There's no external enzymes like ligase or this FFPE repair. Okay, so just to like visually show that, we actually tried a couple different schemes, but in the end we stuck with this 1,200 base pair pieces. So we just amplify the bigger pieces of the genome in 2 different pools, same thing. And instead of this 135-minute procedure, we use the rapid kit, and that's about 10 minutes. Obviously, it depends. Usually, to be honest, it takes me more than 135 minutes to do the ligation kit. It also takes me a little bit more than 10 minutes. If I'm doing one genome, it's feasible to do it in 10 minutes, but anyway, the take-home message is it is faster. There are fewer steps, and it's just simpler to do. And the cool thing about this is that now in March, ONT released this in a 96-well format. So this makes it even easier. And they've even streamlined the procedure more than I did. So really it's just do the RT step, the PCR, you pool the 2 different pools, A and B, together. Then you take 5 microliters of the pool, add it to what's called this barcode adapter plate, or attachment plate, actually. You add 2.5 microliters of water. You incubate the plate for 2 minutes at 30 degrees, 2 minutes at 80 degrees. You add a microliter of a rapid adapter, the motor protein. Incubate that at room temperature for 5 minutes, and then it's ready to, it's not exactly the sequence, actually. You pool, and I've actually, sorry, there's a small steps. Anyway, you do a pooling and a bead cleanup of one pool, and then sequence. So generally it's pretty fast, and it's a bit faster. So, I mean, that's it in a nutshell. We get a little bit more even coverage. These are some of the results from our paper. This is the ARTIC method using the rapid kit. So we are always interested, let's use the rapid kit. It's easier, it's faster for like point of care settings. That would, we knew that was what our aim was, is to make it as simple as possible. So we were interested to see if the ARTIC primers would work. They didn't work that well in our hands with this kit, with the rapid kit, but our primers, the 1200 base pair primers, worked pretty well. So that's what we stuck with. And we call it the midnight set because it was either high noon, like for 1200, or midnight, and midnight just sounded cooler. So that's what we went with. And someone suggested that to us. So yeah, so that's basically, that's the gist of it, to be honest. And ONT has done a lot of work on looking at how this works using the twist synthetic RNA, the COVID genome, and they have, how low, basically the limit of detection is approximately, it's hard to really say this for sure, but about 1,000 viral copies, which is around CT30, it's very similar to the ARTIC method. Everyone sort of has a difficult time sequencing the genome, I think, when you have a really low viral load, like above CT30. So I think it's fairly similar to the ARTIC method in terms of limit of detection, as far as I'm aware. And you can do, 96 genomes in less than 12 hours. So you can fit a whole 96 genomes for the SARS-CoV-2 on one minion flow cell, and get the whole genome within, well, this data shows about 6 to 12 hours. So it's pretty fast. So that's, I think, it. That's the main difference, is just one uses 400 base pair pieces and a ligation kit. Ours is 1200 base pair pieces with the rapid kit. And you could actually use the ligation kit with the 1200 base pair pieces as well. That's fine too. Yeah. I went through that very quickly, but that was just, I think you guys sort of understand some of it, but are there questions? Do you have questions about it? Or what are some of the stumbling blocks that you've gone through, or?
So personally, from my perspective, the main concern is really the cost. So would this mean that it's would cost less than the ARTIC protocol?
Yeah, so it just depends. So we talked about doing a cost breakdown in our paper. It was partly due to time constraints, we didn't do it, but it is, and it depends on if you're using the RBK 004, and I meant to actually put RBK 1110 right here. So cost per genome, if you use the ARTIC method compared to our method, the midnight method is cheaper because it does not use ligase, and it doesn't use the end repair enzymes. So just in terms of which one's cheaper, the midnight method is cheaper. It just depends people always ask, how much per genome? It really depends on how many genomes you're doing. And well, especially where you're in New Zealand, things are a little bit more expensive to bring them in. Depends on where you are, what your currency conversion is, all this stuff. So just head to head, it's really about how much the major cost, or one of the major costs, is the flow cell. So the more genomes you can put on a flow cell, the cheaper it gets. I think ONT has a little, I have it here. They have how much it costs using this RBK1110 kit, the 96 rapid kit, and price per sample. If you're buying flow cells for US $500, so if you're buying them in bulk, $500, the library price for that is 99 US dollars. So price per sample is about $5. So you can sort of, this gives an indication of approximately how much. This doesn't include the cost for doing the reverse transcription or the PCR. So that's an additional cost. It just, the take home message is, I guess, the more samples you can fit on a flow cell, the cheaper it becomes. And there's some base cost always for both methods to doing, they basically use the same method for the reverse transcription and the PCR. So that cost is the same.
So does this mean that instead of purchasing the ARTIC primers from NEB, would be purchasing another kind of primer set?
Yeah, so IDT sells this as a pool, so you can buy it from IDT. I've talked to NEB a little bit about if they're interested. I mean, they can do what they want. They currently, I don't think, sell. We sort of briefly talked about they were maybe interested to look at it and do like a rebalancing like they did for the ARTIC primers. But yeah, you can buy the primers as a pool from IDT. That's the take home. And they cost, I think they cost the same, as far as I understand, as the ARTIC primers. So you can, right now you can buy the ARTIC primers, as far as I know, as a pool from IDT, and you can also buy them from NEB as part of a package which has the other enzymes like the ligase and the FFPE repair. So they sort of bundle that together. Is that, yeah.
Cool.
So,
yeah.
Go ahead.
Just a quick question before I pass it on to the others. I read in the past few months that like when we bought is it the LSK 109 that it's going to be stopped in May? So I don't know if it's still available, because that's basically our only choice for the ARTIC protocol. Is that correct?
So I mean, I think that it will change to the LSK 1110, which is supposedly better anyway. So I think they're just like everything with Oxford Nanopore. Once you buy in, then they change it, and then you have to buy the new thing. But I think even if you go with the ARTIC method, which is great, and you use a ligation kit, they have like an improved kit, which is supposed to actually typically when you start sequencing, you start to see like basically the number of base pairs that you start sequencing starts falling off over time. And that new LSK 1110 kit is supposed to not have that drop off. So it's supposed to just increase your yield. So it's going to get better. And that shouldn't change, even though they changed the number. If you get the new LSK 1110, I think it's not going to change much for your protocols, as far as I know. I haven't used it yet, to be honest.
Cool. Yeah. So I'm sure the others have many other questions, so I'll stop asking now.
Anybody else?
Khalil, mind if I go?
Go ahead, please, Homer.
Okay. Ms. Nikki, my question would be with regards to the wear on the flow cell, because since we're using a longer, we're doing longer reads, have you noticed any, or is there any increase in the wear level of the pores when using your protocol?
I don't think there's much difference between the 400 bases and the 1200 bases. I mean, Nanopore really, this is incredibly short for Nanopore. There, you know, the Nanopore sequencing is really designed to give you these huge, really, really long reads. So the difference between that 400 and 1200, I don't think there's much difference in terms of pore availability or things like that. I think I've heard some people say that if you use the rapid kit with the Midnight protocol, there's a little bit lower yield overall. But if you're sequencing SARS-CoV-2, the genome is so small anyway that that's not such a huge issue. So I think if you use the rapid kit and say 96 genomes, it may take a little bit longer to get the data that you need if you're using the rapid kit versus the ligation kit. But I'd rather spend my time waiting for the sequencing to run rather than being in the lab doing, you know, that's like labor costs that you have to, and there's mistakes that can be made and things like that. So I think in terms of SARS-CoV-2, you may have a slight decreased yield in your flow cell if you use the rapid kit versus the ligation kit, but it's not really a big issue, I think, because the genomes are really small. Does that answer your question?
Yes, yes, that answers my question. Thank you very much.
Yeah, thank you.
And another question would be, what changes did you make in the bioinformatics step to take into account the 1,200 base pair length and the new the new reagents that you've been using?
Yep, that's a good question. So Olin Silander, who I work with, has done most of the bioinformatics, and he's put on like in our paper or in protocols.io, we have a link to, if you're using, it depends on sort of what you're using to do your bioinformatics, but if you're using Rampart or other ones, he has put basically files which you just sort of change the and I'm so bad at this, so he'll explain this better. He can explain this better, but basically you just need to tell the bioinformatics tools that you're using which primers you're using. So we have a file that you can download to modify that. But generally you can use the same methods that you use for the ARTIC, the 400 base pair method, and the 1200 base pair method. You just need to change a few parameters to sort of tell it like, these are the primers that we used.
Okay, thank you.
Yeah. And I can get, if something about the bioinformatics is not clear to you, I can give you his email and he can chat with you about that if you want.
I have a question if no one else has any. Some people are seeing dropout of certain lineages with the ARTIC primers at the moment. Do you have much of an idea of kind of the coverage of different lineages and how it differs to ARTIC?
Yeah, that's a good question. And actually that was, I think Homer had also asked that and I didn't talk about it. So we've done a good amount of work and different ways of doing this, but looking at all of the available sequences and looking at what the major variants of concern are and looking at how our primers are performing in silico, just, you know, seeing how many mismatches there are to the variants of concern, like the major variants of concern, as well as taking just huge amounts of genomes, aligning them, and also checking even rare variants to see how the primers are performing. There are about 5 primers that have some mismatches. There's especially a mismatch for like the P1 variant of concern and primer left 21. This is getting into a lot of detail, but there's like a mismatch right at the 3' the first base on the 3' end of the primer, which usually indicates that one's not going to work. And then another mutation also sort of in the middle of that primer. So we know there's a few primers with certain variants where there are mismatches. I actually, because we're in New Zealand, we don't have a huge amount of COVID samples here. And I haven't looked in depth about, and I'm actually just starting to talk to the people here who are doing the majority of the sequencing here in New Zealand about looking at that more systematically. Short answer is I have about 5 primers right now that I know may be potentially an issue. I've ordered alternatives to sort of fix those mismatches that potentially could be a problem. I've started testing those one by one. So like that P1 variant with that left 21 primer, I tested that. And if you add an additional primer on top that's the correct sequence, you can increase coverage. So what I'm looking at, it's still very, very beta, and it's not a complete picture yet, but I'm sort of, and I don't have a lot of time because I've changed jobs, but I'm trying to look at like a simple fix, which is taking the pool that you have, if you have the midnight pool, and seeing if you can just add a few primers on top that are sort of the fixed primer, which is not the best way to do it, but that's a real life fix, and seeing if that will help coverage. So generally speaking, the only major mutations that are of concern are the P1 in one specific section. But even that, even when I tried it, I did actually get sequence data. I got, it was a dip in coverage, but it wasn't a complete loss of coverage. So you can even though there are some mismatches, it can still work, which actually was surprising. I thought it would drop out completely, but it actually worked okay. Not
yeah,
well.
that's good.
Yeah.
If you have an idea of what's problematic and with which primers, can you kind of infer what you're most likely to have missed if it did drop out completely?
Yeah. So you can sort of look at an alignment, say, maybe if you're in a country where you know this is the variant of concern that's circulating most frequently, you can align the primers and say, this is potentially the region that's going to drop out given the sequence of the genome. So you can have an idea already about the ones that might perform worse with our primers, given the genome. I'm not being very clear, but I hope that answers it. Yeah. You know what I mean?
Yeah, I think I know what you mean. That is helpful.
I mean, but even ones that I thought, like this P1 variant, I thought there's no way this will amplify. But actually it did amplify. It didn't amplify well, but it did amplify, and we got sequence reads. So even things where I thought, oh, this mutation, because it's right at the tip of the primer, that's usually going to cause the most issue. That still didn't completely. It still amplified a little bit. So it's actually these primers are holding up much better than I thought they would, to be honest, in terms of they luckily are not in regions of a lot of variation, and they seem to be, generally speaking, for most of the variants of concern that are out there, they do pretty well in terms of not having a lot of mismatches between the primer and the variants of concern. And the nice thing about the ARTIC, the difference too, I mean, you're using 1/3 fewer primers. So I think I have that even somewhere. I should mention that there are a lot of people who helped on this. I should say that too, which I'll talk about in a second. But here, yeah, the primers anneal to only 4.5% of the genome compared to about like 17% with the ARTIC method. So just based on just numbers wise, it should hopefully perform a little bit better because there's just fewer places where they anneal. Yeah. And I'll come back and say actually that there were a lot of people who helped. So Olin did a lot of the work here. Marketa is a PhD student in our lab, and she did a lot of the work doing some of the sequencing during some of our like outbreaks here in New Zealand. Lab Plus also gave us a few samples to start working with. Our biosafety officer, James Connell, really helped us get all the safety protocols in place, which is another thing that often goes unmentioned, but is really important to make sure that you're working in a safe environment. And also the folks at ESR in Wellington, Joop de Licht and Una Ren, were really helpful as well for this project too. I wanted, before I sort of brushed past that, but it's so important to acknowledge these people because they did a lot of work as well. But yeah, any other questions?
Mitch, do you have questions, Mitch?
Hello, good afternoon. I just like to clarify the PCR amplification. I'm looking at the IDT document for the Midnight Protocol. And then I noticed there's PCR amplification after post-ligation cleanup. Is this dependent on the type of barcoding kit that we are going to use?
No, so you can, basically, it's pretty agnostic in the beginning. So if you have a sample from a patient, you know they're positive, you can use a general reverse transcriptase. That is like the Luna script. That's standard for all of the methods sort of right now. And then you can use either, you know, the ARTIC primers or R primers or even another set of primers to amplify the genome. And then you have amplified material, and you can use that for Illumina sequencing. You can use it for nanopore sequencing. You can use it for anything.
Oh,
if you
okay.
want to use PacBio uses our primers too for their method. They use a slightly different method, and that they use the same primers, but it has a tail.
Also, I'm looking at the wrong document.
Okay, maybe. So
Yeah.
IDT sells as a pool. They're about 20-25 base pair primers to amplify the genome, and there's no sort of tailing on them for the barcoding. So people use all sorts of different methods. I'm mostly just focused on nanopore sequencing, and generally, if you're talking about nanopore sequencing, most people use either the ARTIC method, or maybe I should say maybe also the Midnight method. But there are a number of other primer sets out there that also work. Yeah. So
Yeah, because I noticed in the document from IDT featuring the Midnight Protocol, there's a PCR amplification after the post-ligation cleanup. So like I was wondering, yeah, but
they
because
use
when I look
Illumina
at the rapid kit,
Mhm.
I look at the rapid kit after adapter ligation, you can then sequence the library. So I was wondering if they what is this for? Like, is this for the barcoding step, or
Yeah,
it wasn't clear in the document from IDT?
IDT has something that they call the Lotus. So they amplify the genome using our primers, using the Midnight primers. And then as far as I know, because I haven't looked at that too much, because I haven't done Illumina sequencing, but they use that for Illumina sequencing.
Oh, okay.
So they take like a transposase as well, almost like Nextera. It's called the Lotus kit that they sell. It basically cleaves the DNA and sticks a barcode on. And then they do, as far as I know, because I haven't looked at it too much, I think they do a second PCR to put indexes on. So that's for Illumina sequencing.
Oh,
so.
okay. Sorry, I'm sorry. I did not notice. It's for Illumina.
Yeah, it's for Illumina sequencing. So you can, as long as you amplify, so there's two sort of things we're talking about. One is just amplifying the genome. And then once you have amplified DNA of the genome, you can put that into an Illumina machine, or you can put it onto a nanopore machine. And both methods have different ways of preparing the DNA so that it will go onto their sequencing chemistries. So like Illumina needs a P5 or whatever. I should know P7 and P5, like the adapters.
Can we skip the cleanup after adapter attachment if we use the rapid barcoding kit from Nanopore?
If you have 4 samples or less, you can. I would just add it though anyway, because it's going to improve your yield a bit. And it's pretty rare that you're going to only put 4 samples onto a whole flow cell because that's fairly expensive per sample. You could put it on a flongle, like a mini flow cell. So Nanopore also sells these mini flow cells, which are about 1/10 the price and have only a quarter of the output. So they produce 2 gigabase of data, and they're about $90 US. So that's another alternative for small, SARS-CoV-2 has a very small genome, so that's another alternative is using flongles, these mini flow cells to do this. And so if you have only a couple samples, that might be a good option, price-wise. Yeah.
Yeah, can I go next? Can I ask? Hi, Nikki. I'm Paul. I'm the bioinformatician of ARTIC. So my question is, I think, yeah, you mentioned, I think, beforehand that so we'll just be using essentially the same software from the ARTIC protocol, like the software package, and this midnight protocol will just involve changing maybe some of the parameters or the configuration files to use, but essentially we'll just be using the same software package that we use for ARTIC protocol. Is that correct?
As far as I know, so I think you can. There's actually, I think you can use How are you doing your bioinformatics? Are you using Rampart, the live?
I'm just using the base protocol, the NCOV 2019. I think the most original bioinformatics protocol. So at least for our first sequencing, we'll just be using the baseline. Then maybe in the next sequencing, maybe we can just change a bit, just so we have like a baseline comparison with the most base.
There is, I know there's software online that's web-based, where you can actually pick which primers. It's like, now I'm trying to remember, it's James Ferguson, and I'm trying to remember the name of it. I can look it up really quick, but basically it's supposed to be like a little bit easier than the ARTIC method, and it will take any data and you sort of click. It's a web-based tool, and you say, I'm using the midnight primers or other primers. Their group developed another set of primers, which is a 1,000 base pair. That's John Sebastian Eden. And so that tool can make it easy to do the bioinformatics and to sort of easily click and say, yeah, I'm using this primer set or this primer set. And I can send that to you if you want. But so I'm not a bioinformatician. I really rely on Olin to do all of our bioinformatics. He's actually, I can grab him if you want, and he can answer that question if you want.
Oh, okay, that's great. Thank you.
Yeah.
Any other questions? Cool. So if not, well, thank you so much, Dr. Fried. Any last words, any advice if we try to explore using the Midnight Protocol?
Well, I think given what Nanopore says about on their landing page, if you look, I think, especially if you're looking at using this for people who don't use Nanopore sequencing all the time, it's definitely sort of the easiest method. So if you haven't invested a ton of money, it might be the direction you want to go. I think both work pretty well. I mean, like I said, I feel bad ever saying anything bad about the ARTIC protocol because they're like heroes because they did this so fast and they did a really good job and it works fine. So, I don't feel comfortable criticizing them or anything, but I think the Midnight Protocol with the Rapid Kit is really easy to use, and it's fast and it's cheap. And you can put it on a robot if you have a robot, and I've done it with students who are not very experienced, and they can do it, and it seems pretty simple. So I feel like if you're talking about users who might not have a lot of lab skills, it might be the way to go.
Great.
Yeah.
All right. So, and with that, I think we could end this conversation. Thanks again so much, Dr. Fried. We'll be using this knowledge, and then we'll be spreading this recording so that other labs in the developing world could also consider this protocol. So thank you so much, and have a great day, everyone.
You too. I'll send some links through the email for the bioinformatics tools.
Much appreciated. Thank you so much. Have a great day.
Thank you.
Bye.
Okay. Bye.
Thank you. Bye.
Thank you.
Thank you.